Hello everyone, welcome back to another session of RNA biology. And we were here in the previous class that we were discussing different proteins that are capable of editing the RNA at specific levels, and we know this RNA editing enzymes have a specific loci, that is for recognizing the RNA domain, and also they have specific domain for causing the deaminase function. And we are now going to see more in detail about the tRNA editing, which we kind of looked in the previous class. That is, specific location in the tRNA need to be edited for purpose of enzyme identification and also for the stability of the tRNA molecule. And we also saw this, that the changes in the anticodon part of the tRNA, as you can see here, allows that with minimum tRNA genes you can cater maximum number of the codons. So this is an advantage. So advantage is quite useful or quite lifesaver when it comes to several prokaryotic or several lower order organisms who don't have the luxury of a wide variety of tRNAs. So this GCI conversion is happily be able to cater the arginine codon, because I is neutral. I doesn't have any biasness towards any of the bases. Otherwise, you must have to have a CGU pairing with GCA only. So this problem can be solved, and subsequently the other codons also they were able to cater effectively. Now the outcome is that there are 30 tRNAs are there in total, and 64 triplet codons are there. 61 brings in amino acid, and 3 of them brings no codons or no amino acids, and we call them as stop codons or nonsense codon, which do not bring in any amino acids. So only 30 tRNAs are there. Of course, they are sufficient because we have only 20 amino acids. However, the diversity is useful because you have 61 codons that has to be catered by 30 tRNAs. So this editing capability allow this variation to be brought in effectively. So you can see the codon preference table. This is the first base of the codons, like U, this is written here, and this is the second letter U, here it is U, and this is the third letter. So U, U and U, it is written U, and then comes second UUC, U first letter, U second letter, and this is C, UUC, and first letter, second letter, then A, UUA and UUG. So like that it keep varying, and accordingly you will have different phenylalanine, leucine, like that it keep varying. So this discovery was done by one of the scientists of Indian origin, and his name is Hargobind Khurana. So he also received Nobel Prize for the discovery of the genetic code. And so all bases are vulnerable to changes, and this slide you may have seen it in the one of the earlier classes also, where we are discussing about the RNA modifications or the base modifications. Uridine, cytidine, adenosine and guanosine, all these bases can change into ribothiamidine, dihydrouridine, pseudouridine, 4-thiouridine, etc. Like that, cytidine also can be changed into 3-methylcytidine, 5-methylcytidine, etc. So adenosine also can change into inosine, N-methyl adenosine and N-isopentenyl adenosine, etc. And guanosine can change into 7-methylguanosine, which we know, we are all familiar with because it is used for 5' cap. And then also Q-inosine and also Viosine. So these modifications are in different tRNAs and they can influence the functionality of the stability of the RNA molecule. So such modifications are not done random, they are done at specific basis and they are done with a purpose. So we should understand the changes, each and every changes that is occurring in individual basis are having one or the other purpose. And let us see some example of CU editing, that is cytidine or cytosine to uridine, CU editing. And we know plenty of enzymes we have seen so far, it is the same slide repeating, and now we are going to deal with a RNA editing enzyme called ApoBec. ApoBec is a well studied molecule, it has got huge implications, it is important in RNA editing and it also helps in a lot of other unrelated functions also, in the sense it is made use of by various viruses for its own benefit, etc. So we will see them with some example in detail. So what is ApoBec? Apolipoprotein B mRNA editing enzyme catalytic polypeptide 1-like, that is the full form of ApoBec, and some portions have been taken, that is highlighted in red color, ApoBEC, and 1-like is given this number. So this functions with ACF. What is ACF? ApoBec complementing factor. So ApoBec is an RNA editing, like earlier also I have told you that some RNA editing molecule will not have RNA recognition domain, they have the deaminase domain. So they will collaborate, they will always get along with another protein which has the RNA recognizing capacity. ApoBec is one of them. So you see the example ACF here. How does it function? So what is the significance of CU editing? ApoBec 1 editing in ApoB. Let us see how is it happening. CU editing is what is happening, and which codon it is editing? CAA is now changing into UAA. And we know, we have seen another example earlier where the liver and intestine was functioning, a variation in the apolipoprotein B. And here you are seeing another example is CAA become UAA. UAA is a stop codon. You know, there are 3 stop codons are there. UAA is one of them. And what happened? This ApoB can be of 2 different molecular weight because of introduction of this stop codon in certain cell type. So ApoB 100 and ApoB 48. Let us see, this is the structure of the apolipoprotein B. So codon 1 to codon 4564, and you have a stop codon introduced here 2153. So now usually in liver and intestine this is how they work, and in liver it is unedited and you end up getting a full-fledged protein apolipoprotein B, and which has got 4563 amino acid, and it functions in the transport of cholesterol in the blood. That is very important function in the liver. Whereas in intestine you have an unedited RNA usually produced which undergoes editing with the help of ApoBec, and this will introduce a stop codon, and this stop codon will stop the translation. So the mRNA will terminate its translation prematurely, and this apolipoprotein B is apolipoprotein B48, which has got around 2152 amino acids, and its function is totally different. Function in the absorption of lipids from the intestine, which is quite handy because intestine is a place where the nutrition, food what you take inside should be absorbed effectively. So in both the tissues, liver and intestine, the derivatives, one is a full length, another is a truncated version, both have got an effective function. So now you see the example, RNA editing is not only able to bring in 2 different proteins, of course they have common 5 prime regions or the N-terminus end of the protein they have common. However, what is important is with one gene, it is able to perform the task of 2 genes or 2 proteins in 2 different tissues. So it is very advantageous for the organism. Now let us see another angle of functioning of the CU editing. So the innate immunity to retroviruses with cytoplasmic apobic. Apobic can be nuclear as well as cytoplasmic. Okay, so RNA can be edited in both the places depending upon which RNA you are talking about. And apobic is associated with cytoplasmic P bodies. And the adaptive immunity by AID. What is AID? Activation induced cytidine deaminase. So cytidine deaminase function, because cytidine, if it is deaminated, it will become uridine. This function is adaptive immunity related. So this adaptive immunity by AID is something which is very important for us to go further. And of course there is a nuclear apobic also. We are dealing with cytoplasmic apobic. Let us see what is the relevance. So apobic 3 and AID, they interact together because we have already seen here that it functions along with other factors, other proteins, and adaptive immunity is caused by activation induced cytidine deaminase, that protein. So apobic and AID have got a good interaction or good interrelationship. So if one example such as HIV virus, which cause the AIDS, and this HIV, once it enters the cell, and we call it as HIV virion, because the HIV virion is the one which is capable of coming out of the cell as a fully infective particle. So it can stay in a dormant state also. So what you can see, this is a typical HIV particle where it has got its own genome and also it has got an envelope, etc. And then virus also have specific proteins which it produce because which is not available from the host. A lot of virus functions are dependent on the host protein, host enzyme, host machinery, but a host will not supply everything what a virus needs. So virus will carry only those things. Like you can see a simple example. If you are going to write an exam, they will provide the chair or a table or a bench to sit and write. They will also provide you paper to write the exam. They will not provide you dress. They will not provide you pen. They will not provide you, if you want a pencil or eraser. No exam hall will provide. You have to carry. But paper you don't have to carry. Paper they will give, because your paper is not trustable, because someone can manipulate it. But pen you don't. You cannot manipulate a pen. So this is what you should understand. So virus, if it need, sometimes if you are calling a skilled worker, a carpenter to your home, you need to provide him a place and tool only. Carpenter will bring the tools which is required for his work. You don't have to supply tools to him, because without those tools carpenter cannot work. So same logic applies when virus comes. Virus makes use of lot of things from the host, including some proteins such as apobic, but very unique proteins which no host can provide, virus brings in, just like a carpenter is bringing a tool or you carry pen for writing exam, like that. So VIF is a protein which is viral specific, and what happens is that if there is a specific loci is there, it can convert into A3 protein can convert into A3G, that is it can get converted into A3F, and alanine become glycine or alanine become phenylalanine. So such kind of editing is possible with the help of when there is apobic protein is present in the cytoplasm. And when you are talking about AID, immunoglobulin locus, because we all know that when there is an infection, it always brings in changes at the hemoglobin. Normally when you have an infection, your blood serum contains plenty of antibodies which can target, but their number is less. Say for example, if your blood serum has 10,000 different types of antibodies are there, which can target pretty much any known virus in your body, but their number is low. So as soon as you get an infection, one of them, these innate antibodies will go and recognize, but they cannot conquer, they cannot completely eliminate the infection. But this small percentage which could detect the host will be amplified. And some normally in the immune cell, the VDJ, variable domain mutations are very much possible, the variable region, so that there is a concrete backbone of an antibody and the variable loci which will be the pairing portion, which can bring in changes. And if these changes are able to target a virus, then that particular change bearing antibody will be expanded. It is just like you are running a shop and you don't know what people need it. Once you produce a product and there is demand, you kept 10 products, but 3 products have got heavy demand. Then what will you do? You will start producing this 3 product more, and you will reduce the production or stop the production of the 7 other items. Like that, if there is a antibody which detected the host, then the organism will expand it. So that is why antibodies usually have the variable region. And this is a notation W, R and C. C we all know, it is cytidine, and W and R are code for nucleic acid. R stands for purine and W stands for A or T or A or U in that case. So in this conversion, WRC to WRU is very important for the functioning of the AID, and this is basically the switch regions of the antibody. For your convenience, we will show that this is the IUPAC nucleotide code A, C, G and T or U, and that is the the nitrogen base, that is the nucleotide, and R stands for A or G, which is nothing but purine, and Y stands for pyrimidine, that is C or T, so on and so forth. You can see it, and that is the code which is used in the previous slide. Now coming back to the virus, what happens is in a producer cell, usually the virus gets integrated. The viral, because HIV is a RNA virus and it converts itself into DNA and integrates into the genome of the host, and that is why we call it as retrovirus, and it is staying in the genome, we call it as a provirus. So provirus basically means it is a virus, but it is not a virus. It's genetically it is a virus, and that once gets converted into RNA, that RNA can get converted into protein. And I think HIV genome is around 30,000 bases. It is much higher than any other virus. Usually virus will have around 10 to 12,000 base only, but HIV genome is little bigger. So what happens when this viral RNA is produced? It can happily produce the viral protein, viral coat proteins, etc., and it can happily get itself converted into a functional virus, come out of the cell and infect other tissue, like you can keep spreading. Like virus, once it is entered into the genome, it can transcribe and come out and make fresh virus anytime as the virus wish. If your immune system is strong and you eliminated all the virus, then HIV will keep quiet. It will stay dormant in your genome. So whenever your immunity is little low, it will start producing from the genome. So it has the talent. So what we should understand, the apobec is from your body, okay, and apobec is a very interesting situation, because normally if apobec is produced, it has to do its job, that is causing the cytidine deamination. Usually it undergoes degradation if VIF protein is present. VIF protein is from the virus, it is sent for degradation. Otherwise, if it is not degraded, what it will do? It will incorporate into the viral particles and it can come out and get into a host cell, and it can carry virus along with it, and it can sometimes, if there is a host derived RNase is an RNase enzyme that can specifically degrade the RNA part of a DNA RNA hybrid. If RNase H etc. comes into action, it will be degraded. But what the system will do is that it will make use of this apobec, and it can decide whether it should be sent for degradation or whether it should reverse transcribe and go into the host or integrate into the host. So if CCT or CCU, what you are talking, if it is there, CCT, if it gets converted into UCT because of the apobec, because we know apobec is a cytidine deaminase, it gets converted into UCT, it can have completely different meaning for getting into. If it got converted into UCT, it is capable of going into the genome, but if it did not, a part of it can also undergo the degradation, and even this capable of integration also can mark into degradation. So what we should understand, that this is a strategy. It can easily make use of apobec, either mark the apobec for degradation while in presence of VIF protein, and if VIF protein is not there, the virus is not producing it, then it can, the apobec can carry along with the virus and go into the host cell. Even if host cell is not providing apobec, it can happily make use of it. And those who are interested can read this retroviral restriction by apobec proteins. It's a very interesting article because this will have more in-depth knowledge and subject to understand, because if I start explaining, it will take much longer time to understand the entire dynamics of apobec protein, and also understand apobec proteins are important in the cellular homeostasis of various other tissue types other than this immune related function, because they are important even in the regeneration biology also. So this is a table, it's a very elaborate table, that RNA editing involving A to I conversion and the known substrates and the functional consequences. Like you can see in mammals, Drosophila, hepatitis virus, etc., etc. And the transcript basically we are meaning the gene, that is GLU-RB, GLU-RC, like that. And what is the effect of editing? These are all single letter code for amino acid. Q to R means glutamine to arginine. R to G means arginine to glycine. Like that it goes, because 20 amino acids also have got a single letter code. And you also see what is the functional consequences, because decreases the calcium ion permeability in one case. Like that you can see the whole list, because showing this list is not for memorizing purpose. Showing this list is for you to have an idea how many genes in how many organisms it is. Even in Caenorhabditis elegans and measles virus, polyomavirus, etc. also make use of this RNA editing. So RNA editing is not a chance event. RNA editing is a planned, purposeful event, because of which various viruses and various other eukaryotic genes are able to make a proper living and a proper way of functioning in the host. So this is what you should understand in detail about the RNA editing. Now we can understand more in detail about RNA splicing, RNA export and RNA decay, because this is also part of this section. And in this section we may get into some topics which we may have already covered little bit, but understand this touching upon is necessary so that you can, first of all, you can recap little bit, and then it also can bring in the context relevance or the importance of the topic that is being introduced in this section. So don't think that some topics are being repeated, because they are intentionally put in, but I have tried the best way possible to avoid the feeling of repetition, because they are basically for memorizing the previous topic. So we know the pre-mRNA processing have got capping, splicing, polyase signal, cleavage and polyadenylation, etc. This happens from the DNA. Now when it comes to RNA splicing, we know it comes from the DNA, and it has got exon 1, exon 2, exon 3, and also have got 2 introns, and the spliceosomal machinery works on the primary transcript and or the pre-mRNA and causes the production of the mature transcript. We know this is basically the fundamental principle, and we also know that this has fetched the Nobel Prize for the discovery of introns by Richard Roberts and Philip Sharp, and that's why we call them as split genes. And exons and introns are very important, not as an intruders, or introns are not really intruders because they are very important for the normal functioning. And we also need to understand that introns and exons hold some information about, because once the exons are fused, you are technically having no idea whether base number this to this, say base number 1 to 200 was exon 1, 201 to 712 was exon 2. We have no idea, absolutely no idea. That's what we assume. But the truth is, no. There are signatures on the RNA, and there are even proteins that bind onto individual exons and give clue to the system that this is the boundary of an exon. And you may wonder why that is needed. That is definitely needed because that is part of marking whether or not a given RNA should be sent for degradation or not. So we need to understand more in detail about that, and we will see them one by one, because each exon or each intron and the exon intron boundary, even in the processed RNA or the post processed RNA or the spliced RNA, this boundary have got relevance. So who is providing this relevance, etc., we will see them one by one in the next class. Thank you.